can find all what you need regarding the pharmaceutical sciences. I am Dr. Mina Samir, R&D Formulation Section Head, and I will be your companion today for this webinar about the fundamentals or introduction to solid dosage form formulation process. Starting our webinar today, we will be talking about main 6 points in our agenda. First, a definition of a dosage form and overview of solid dosage forms, as solid dosage forms are our main concern of this webinar. Then number 2, the role of formulator, including main 3 aspects, which are selecting the innovator product that will guide the development process, then performing a pre-formulation study, and then the development or formulation cycle. Third, we will have an overview about the main excipients used in solid dosage forms, their traits and their roles. Fourth, preparation techniques of solid dosage forms. Then we will have an overview or a glance about the compression process and the practical case study to apply what we have learned through our webinar. Hope this webinar will be informative for you, and note that this is an interactive webinar, so feel free to share your questions, to share your insights, and also I will be sharing some questions throughout the webinar. So I hope we will be interacting well today in order to get the best outcome of this webinar. Starting on the first point, what is dosage form? According to USP, which is the United States Pharmacopoeia, a dosage form is a combination between the API or the active pharmaceutical ingredient, which is responsible for exerting the pharmacological effect in the body, and often excipients. Excipients are inactive ingredients. They do not have a pharmacological effect in the body, but they are essential in dosage forms, and especially in solid dosage forms, our concern tonight. A dosage form will facilitate dosing, administration, and delivery of the medicine to the patient, as the dosage form is designed in order to facilitate for the patient to gain his required dose, and also to be administered in a safe and effective way, and also to help the API to reach its target place in the body, to be absorbed well, and to do its effect in the body. So in other words, dosage form will ensure correct dosing. If a patient is targeted to have 10 milligrams of certain API, the dose will help him achieve this target, will enhance the drug stability, and improve patient compliance. So main types of solid dosage forms, we will be talking about tablets in particular. So we have film-coated tablets, effervescent tablets, chewable tablets, sublingual tablets, orally disintegrating, and also the modified release tablets like delayed release and extended release. First, solid dosage forms, and especially tablets, are the most common types of dosage forms. They have many advantages as they are of lowest cost. They are very cheap, so they are easy and effective to be manufactured. They are easily to be transported. There is no fear of breaking a glass or something, and also they are the most stable. They don't have high moisture content, so they are not liable to microbial growth. So solid dosage forms are the most common dosage forms in the pharmaceutical industry. But after all these advantages of solid dosage forms, there are some drawbacks or disadvantages, and the main one of them is the difficulty of swallowing for the tablets, especially for the elder people and the children. And even after this disadvantage is discussed, you will find that many solid dosage forms are here to encounter for this disadvantage, which we'll talk about now in the types of solid dosage forms. First, the film-coated tablet, and the film-coated tablet is the most simple form of tablets. Powder is compressed and is coated for acceptance or appeal for the patient, and it is supplied carrying its active ingredient, and it is swallowed by the patient in order to be disintegrating, and API is dissolving in the GIT and absorbed to perform its action. Other types of solid dosage forms that may encounter the disadvantage of difficulty in swallowing, starting by the effervescent tablets. Effervescent tablets are tablets that are intended to be drunk. Here, the tablet contains weak acid like citric acid, for example, and sodium bicarbonate, an alkaline component. And the tablet is placed in water where the acid and base will react together, releasing carbon dioxide, and the API is dissolved or dispersed in the water and will be drunk instead of swallowing a compact tablet. That's an important way to get over the difficulty of swallowing for some active ingredients in tablets. Another type of tablets is the chewable tablet, which is crushed in the mouth into small pieces that will be easy to swallow, and then these crushed parts will start disintegrating and releasing their content of API in the GIT, and here the release of the API starts in GIT and absorption occurs. So the API will reach to the bloodstream and start its action. Chewable tablets are easy to be swallowed, and they have a pleasant taste, so they are not offensive to the patient, and are another way of overcoming the difficulty of swallowing for tablets or solid dosage forms. Another type is sublingual tablets, which is a tablet that is placed below the tongue, and here the tablets fast disintegrate under the tongue, and the absorption of the API starts from the mouth, from the oral mucosa under the tongue, and it has a very fast onset of action as it goes directly to the bloodstream from the mouth, and also will overcome the disadvantage of difficulty of swallowing. And finally, orally disintegrating tablets, where the tablet will have a fast melt action in the mouth. It starts to disintegrate rapidly in the mouth, and a dispersion of the API is created that will be swallowed, and its absorption will start in the GIT. Also, orally disintegrating tablets will have a pleasant taste, so they will not be offensive to the patient. It will be acceptable for him, and it will overcome the difficulty of swallowing. The final type of the solid dosage forms or the tablets are the modified release tablets, which are further classified into two types, which are delayed release tablets and extended release tablets. Can someone tell us the difference between a delayed release tablet and an extended release tablet? Both are modified release, but there is a difference between them. Can anybody tell us the difference?
I think, okay,
Yes.
I think the extended release is about to make the drug to release the API for example, 12 hours or 8 hours or 24 hours. But the delayed release is about to delay the site of absorption of the drug.
Yes, perfect, perfect. Delayed release tablet. An example, the most common example are enteric coated tablets, where the site of action of the tablet is controlled. The tablet does not release its content of the API until it reaches, for example, the alkaline pH of the intestines, where the coat starts to disintegrate and tablets open to release its content of the API. That's the delayed release tablet. And this is always to protect the API from the acidity of the stomach, for example. And in other cases, we are protecting the stomach, as the API will cause irritation for the stomach. So we control the release of the tablet to be delayed until the intestines. While the extended release, as you said, is a tablet that is designed to release its content of the API over a long period of time, maybe 12 hours, maybe up to 24 hours, like you said. And here it can retain the level of the API in the blood at the required level for long period of time, and this will help to reduce the dosing frequency to the patient to increase his compliance, as instead of taking 2 or 3 pills per day, only 1 pill will be taking the job for the whole day. So this was a quick glance over the different types of tablets, and then moving forward for the role of formulation regarding tablets or solid dosage forms. After registration of a new product to be produced in the pharmaceutical factory, the formulation department takes over for the development process, and of course, the methodology will be doing his part for controlling the quality attributes of the product. But we are here talking about the formulation, where there are main 3 aspects or points we will be talking about today. First is the innovator selection, second are pre-formulation studies, and third, the formulation cycle. Starting with the innovator selection, first we must know what is an innovator. Definition of innovator is that it is an original, first of its kind pharmaceutical product containing specific active ingredient that has not been previously used in any other drug. Which means for the company who invented for the first time an API like, for example, paracetamol, and created the 500 milligrams paracetamol tablet, that is the innovator or brand or reference product for any paracetamol 500 milligrams tablet that will be ever produced again. So innovator products are typically protected by patents and data exclusivity periods. So they will be produced for a period of time only by the company who invented them. And then after this patency period is over, other companies are allowed to create generic or similar products to the innovator product. So the innovator product or the reference product serves as a standard against which a proposed generic or biosimilar version must demonstrate similarity or equivalence. In our case, if we are attempting to develop a product containing X API in a certain concentration, then the first step is to identify the innovator or brand or reference product that will be leading the process of development during the formulation cycle. So the first important point that must be identified about a brand or an innovator product is that it must be marketed in a country that is considered a reference country. If you access to the EDA or Egyptian Drug Authority site, you will find a list on this site that mentions the countries that can be considered as reference countries. For our brand to be selected, it must be marketed on one of those reference countries, for example, USA or Canada, many European countries like UK, Germany, and Italy, and also Japan from Asia. These countries are, as mentioned, identified by the EDA and must be marketing the product that will be selected as an innovator. Does any product marketed in these reference countries can considered as an innovator? That's another question that I need to get the answer from you. Is any product marketed in USA can be considered as an innovator product? Does anybody know about that? Do we have another volunteer? Fine, I will proceed with this point. Not every product marketed in the reference countries is considered as an innovator, but the product must have certain criteria to be further selected as an innovator. And to help you identify these criteria, here we mention a list of the most important sites that are used to select an innovator product or identify an innovator product. First site is the Orange Book, which is a publication by the United States Food and Drug Administration. And here all the products that are marketed in the USA are present. You can find them listed in the Orange Book site. And here you will find that each product is mentioned to be RLD, like reference listed drug, or RS, like reference standard or not. If the product is mentioned to be RLD or RS, then it can be used as an innovator or reference product. But if not mentioned to be one of those two, then it cannot be used as an innovator product. Other site like Medicine and Healthcare Product Regulatory Agency in the United Kingdom. Here the products marketed in the UK are mentioned, and also there are several documents regarding these products are published on the site, and in these publications or documents, you will be able to identify the product that can be considered as a reference product or not. Another important site is the French National Agency for Safety of Medicines and Health Products. That is site for products marketed in France. Also, European Medicine Agency for countries of Europe, and Pharmaceutical and Medical Devices Agency in Japan. That's the site dedicated for products of Japan. And finally, Heads of Medicine Agencies, which is a collective site where you can find different products marketed in Europe, and you can identify some of these products as innovators and some are not innovators.
Dr. Mina, Dr.
Yeah.
Mahmoud have a question.
Yes,
Can you ask, Doctor?
sure.
Dr. Mina, may I ask what is the difference between RLD and RS?
Dr.
Sorry,
Sam,
the voice is very far. I can't
Yeah.
hear well.
What is the difference between RLD and RS?
They are both the same. They are RLD means reference listed drug. RS means reference standard. And if any of them, one of them or both of them are marked in front of a product, it can be considered as a reference product. So if you access the Orange Book and start searching for a certain API, like for example, paracetamol, the example we talked about, you will find each product containing paracetamol marked in the USA. And you will find two columns. One is named as RLD, reference listed, RLD reference listed standard, or RS reference standard. And both of them means that this product is an innovator product. You don't have to find the two of them marked. Only one of them will mean that this is a reference product. Also, both of them is meaning that this is a reference product.
Thank you,
That's
Doctor.
right. It's okay.
Yes.
Does any other have any
Doctor,
questions?
yes. We have another question?
Yes.
Doctor, can you ask?
Yes.
Dr. Mubashir, Doctor, you can open the mic and ask your question. Hello, Dr. Mubashir. Unmute and ask your question. We can proceed, Dr. Mina, until Dr. Mubashir ask his question.
Okay. Feel free at any time to ask your questions. But now we are moving to the next step of the formulation rule. After identifying the innovator product that will be guiding our development process, we will have to perform a pre-formulation study. And during pre-formulation, we are trying to understand each aspect about three main components. First is the API, the active pharmaceutical ingredient. Second is the excipients used in the dosage form. And third, the innovator product itself. First, according to ICH, which is the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, the ICH states that the physicochemical properties of drug substance can influence the performance of the drug product and its manufacturability, and these aspects should be identified and discussed. Stated examples are like solubility, water content, and particle size of the API. About that, solubility of the API must be understood as understanding this process or this feature will help us for better development process. If I know that this product has a slow rate of solubility, then I may be adding a solubilizing agent or another excipient that may help the API to release. And solubility can be understood by understanding the BCS classification, biopharmaceutical classification system, which is depending on two main pillars, which are solubility and permeability. Products are categorized into 4 categories of high solubility, high permeability, high solubility, low permeability, or low solubility, high permeability, and finally low solubility, low permeability. Understanding these characters will help you properly develop the formula, as you have a background of the traits or the characters of your API. Also, water content is required to be understood. For example, if you have a product that is monohydrate or dihydrate equivalents, you will have to calculate the proper amount of API to be added per tablet in order to deliver the required dose to the patient, as 10 milligrams of the API are not equivalent to 10 milligrams of the API monohydrate. Here you have to calculate and adjust the amount of API per tablet in order to get the required dose per tablet. Third aspect that we will be briefly discussing is the particle size. And the particle size has a great influence on the release profile of the API. For example, increased particle size will mean there is a slower rate of dissolution and release of API. On the contrary, a fine or small particle size will provide high surface area, so increased rate of solubility. So understanding aspects or traits of the API, like the 3 we mentioned before, and many other aspects also are present. Understanding these aspects will help you properly during the development process of your product. Second are the excipients, and also according to ICH guidelines, it states that the excipients chosen, their concentration and characteristics can influence the drug product performance or manufacturability. Also, compatibility of excipients with the API and other excipients should be established. Excipients are placed in our dosage form in order to enhance its manufacturing process, enhance its stability over time, and also enhance its performance in the body. So understanding the traits of our excipients will help us develop a proper process for a proper dosage form. Also, understanding about the stability of the excipients and their compatibility together or with the API will help you for a faster development process instead of too much trials and errors. Ability of excipients to provide their intended functionality and to perform throughout the intended drug product shelf life should be demonstrated. And for example, if we are formulating a capsule that contains a disintegrant, like for example, cross-carbonyl sodium, which is stated to have its efficiency or its work done in the capsules at a concentration of not less than 10%, then I must add this disintegrant in the capsule in the required concentration in order to perform its role. It's not acceptable to add the disintegrant at a low concentration of 3%, for example, and to wonder why the formula is not performing its function in a proper way. So, understanding about the excipients and their required concentrations or favorable conditions to perform their function is essential for a proper development process. Another example is not related to solid dosage forms, but we can discuss it briefly. For example, benzyl alcohol, which is used as preservative, best provides its action in pH less than 5. If pH is higher than this value, then the preservative will not be performing its function properly. So during the preformulation study, there must be proper understanding of the excipients, the rules, the required action, their compatibility to each other, the concentrations at which they will give you their best performance, and also the favorable conditions for them in order to perform. Studying those traits or characters of the excipients will help you provide your formulation process more efficiently and more rapidly. Moving forward to the third aspect to be studied during the preformulation study, which is the innovator itself. After selecting the product that will be considered as my innovator or reference product, there must be a proper understanding about this product that will be guiding my development process, understanding its qualitative composition. Each ingredient that is added in this product must be added to my generic or similar product, as my target is to create a product that is exactly the same as the innovator product or reference product. So the same excipients or ingredients used in the product, innovator product, will be used in our development. If any of the ingredients is mentioned quantitatively, which means that quantity per tablet is mentioned in any publication, then I must be sticking to this concentration during the development of my product. For example, many tablets or other formulations that contain an antioxidant or a preservative, the quantity of those are always mentioned on the box of the product or on the publications in the sites. And here I am strictly following these concentrations in order to get a product similar to the innovator or brand product. There are many sites that you can find data about the products that are considered as innovators, like, for example, FDA public assessment reports, which are containing data about the products that are marketed in the USA, EMA public assessment reports for products marketed in Europe, also RX List, drugs.com, and EMC. All are valuable resources that you will find data about the innovative product selected for leading the development process by your lab, and understanding properly about the characters of this innovator will help you produce a generic or a similar product that have the same traits or characters like the innovator product. Third rule for a formulation lab: after selecting the innovator product and after performing the pre-formulation study, including understanding about the API, excipients, and also the innovator or reference product, then starts the formulation cycle. After understanding these characters, a proposed formula is placed. Proposed formula based on the qualitative composition of the innovator, which contains, for example, avicel, cross-carbamylose sodium, povidone, whatever ingredients are mentioned to be present in the innovator. Same ingredients are used for our development, and according to experience and some other resources, like, for example, Handbook of Pharmaceutical Excipients or FDA excipients database, will help you to place a suggestion or a proposal for each ingredient is present in what percent or what amount in your proposed formula. And here the proposed formula is prepared as a trial, and the first step is physical assessment. If we are preparing a tablet in the lab and we are trying to perform physical assessment for the granulation, sealing, and compression process, Can anyone suggest what physical assessment can be performed during this process? What I can do as a physical assessment during granulation, sealing, and compression? Do we have an insight here? No?
Can you repeat the question, please?
Someone is trying to say something?
Can you repeat the question, please?
If I am performing a preparation process of granulation, sealing, and compression, what are the physical assessments that can be done to this process?
We can like hardness, size, weight, resolution, texture, color.
Okay, great. Do we have
To
other
ensure the consistent quality, safety, and performance, ensuring that the
Okay,
You are looking for tablet hardness or bulk and tab or what? Yeah, disintegration. There are many aspects. I don't know what you
there are
are
many
looking for
aspects. Yes,
exactly.
we
Yeah.
are talking about examples. If you have an example in mind, we are just discussing examples. We are not mentioning all the aspects. We cannot do that in just a webinar. Right, like what you said, physical assessment maybe also, for example, during a sieving process, if a product has a low melting point and it starts to melt during sieving, that's a physical assessment. The product is not performing in a proper way for the steps I proposed. Then I have to change, for example, the technique of milling in order to get properly milled granules without melting, or I may change from granulation to direct compression if this is possible. During compression, there may be bad flowing powder, so the dyes are not properly filling and weight variation occurs. All these are examples of the physical assessment, and I cannot proceed to the next step if these physical aspects are not acceptable. If I am getting a tablet that has a proper assay and proper dissolution profile, but is not flowing properly during compression, then this is not an acceptable formula, and I cannot take this formula for a scale-up process in the production. I have to assess my physical characters of the product and make sure that is acceptable physically, and then proceed to the next step. If, like, for example, the product is showing bad flowability during compression, then I may change to a granulating process. For example, I may use another diluent that has better flowability than the diluent I have in my formula. Maybe use a glidant like Aerozil or increase its %. So there are many modifications that can be done, and I am not proceeding towards the chemical assessment without obtaining successful or proper physical characters of the product. So after physical assessment, and maybe we need a reformulation or a modification in the formula itself or in the preparation method, then if we get a passing physical assessment, then we go for the chemical assessment. Chemical assessment is assessing different aspects against the innovator product for sure, like, for example, assay. And for the tablets, the most important is the dissolution profile. Dissolution profile, in brief, is the rate of release of the API from the tablet in a certain fluid after a certain period of time at certain conditions. For example, if I am testing my dissolution for a tablet in HCl, 0.1 normal HCl, which is similar to the acidic pH of the stomach at a temperature of 37.5 degrees, also similar to the temperature of the human body, using an impeller or using a paddle at certain speed, for example, 50 RPM. then I will be comparing my product to the innovator product. If the innovator product has a fast release rate of the API, for example, 90% release after 15 minutes, and my release profile is slower, 50% only after the 15 minutes, then my product needs modification. I need to change the formula by increasing, for example, the disintegrant, maybe just modifying the compression process to compress at a lower hardness that allows faster disintegration of the tablet, or maybe change the process from a granulation process to direct compression. There are many modifications that can be done, and then chemical assessment for the product again. And on the contrary, if the API of the innovator product has a slow onset of release, only 50 or 60% released after 15 minutes, and my product has a fast onset of release, then I must adjust my release rate to be similar to that of the innovator. So it's not a competition. It's not, if my release rate is faster than the innovator, that does not mean I am better than the innovator. I must be sticking to the release rate or the release profile of the innovator over time. Here we carry out analysis for several time intervals, like 10 minutes, 15, 20, 30, 45, and sometimes 60 minutes. And in each interval, the release rate or the dissolved amount of the API from the innovator must be similar to the release rate of my generic or similar product. If not, modifications in formula, modifications in the preparation process are performed in order to get a similar formula to the innovator. After getting a formula that has passing physical assessment and passing chemical assessment, we move to a stability study. In brief, a stability study is performed in order to make sure that my product has the same traits or characters starting from its first day and ending in its shelf life of 2 or 3 minutes, or maybe years, 2 or 3 years, or maybe more. So I am making sure that the powder will have its characters unchanged from day one of production to the last day of its shelf life. During the stability study, we subject the product to exaggerated factors or characters of temperature and humidity for 6 months. That is an accelerated stability study. Of course, there are other types of stability studies, but the accelerated stability study is the most common, as it saves time. For after these 6 months, we may be starting over the formulation again. So an accelerated study is always the best choice to be done. In some cases, we may go for an intermediate stability study or an actual time stability study, but our first line is the accelerated stability study. Here, the product may undergo physical or chemical failures during the stability study. Maybe a tablet changes its color or its surface. Maybe its texture becomes rough, for example, or maybe it has failing chemical characters like a failing assay or a changed release rate of the API other than the initial release rate in the first day of production. And here, a reformulation may be done starting from a very simple step like changing the packaging material so it has a higher protection, so the product keep its traits without changing, or maybe we go to change the ingredients or the preparation method. For example, if my tablet has an increased amount of moisture content in the beginning, it may affect its stability, so we go for a more effective drying process during the preparation. Maybe it requires an increased amount of disintegrant in order to keep its release profile over time without change. So a stability study is assessed physically and chemically to make sure that the product has not undergone any change over time. So it has its characters retained from the first day of production to last day of its shelf life. If the product has passed the stability study, Then the product is released and it will go to under scale to perform scale up in production, and the lab trials are produced at larger scale, and production batches are starting to appear. So formulation cycle in summary, physical assessment followed by chemical assessment, and then a stability study, which includes repeating both physical and chemical assessments over time. And if a product passes all these assessments, then product is released and goes to the next step in production. So that was the role of formulation, selecting the innovator product, performing our pre-formulation studies, and then the formulation cycle. And of course, in this webinar, talking about the role of the formulation requires so much time in order to cover all the aspects. Like the doctor said, if we are talking about the physical assessment of the products, we will need so much time in order to cover all the aspects. So here in the webinar, our time is limited. We take just a glance over the process. If you are willing to understand more about the role of the formulation, you can continue learning with us, as our course is present on our Pharma course website, which is covering this process in more deep details. Moving forward, we will be talking about the excipients used in solid dosage forms in brief, starting by the definition of an excipient. According to USP, again, United States Pharmacopoeia, excipient is an ingredient of a dosage form other than the API. So in other terms, excipient is synonym to inactive ingredient. It does not have a pharmacological effect inside the body, but it helps the dosage form to perform its role in the body. Excipients will aid in manufacturing, will support the product stability and enhance its bioavailability, and also will improve the patient acceptance to the dosage form. Talking about the ideal properties of an excipient, they must be stable, safe, and inert. Stable, as the excipient's role is to keep the product stable over time, so the excipient itself must be stable. That is easily to understand. But talking about the safety and inertness of an excipient, here we must mention an important point, that no excipient is absolutely safe or absolutely inert. So can someone share with us an example for an excipient that is very well known to be inert, but have some interactions with a certain API? Does anyone know an example for that?
I think mannitol may have a Maillard reaction in some cases.
Yes, great. So mannitol is widely used in many formulations, but for certain APIs, there may exhibit an interaction, right? Do we have another example? Great, thank you. Do we have another example?
I have one in mind, but it's related to nitrosamines.
Yes.
I believe it's the povidone family, mainly the cospovidone, has a risk of inducing or increasing the nitrosamines risk in the tablet, especially in the
As a
yeah.
general rule, or when interacting with another ingredient?
No, it's a general rule, especially in the presence of moisture or wet granulation.
Okay.
The
So this
induction of
can be
water.
considered as a safety aspect, right?
Yes.
Okay,
Yes.
we can put this as an example for an excipient that is widely used, but it has some safety aspects that can be considered not to be added at a certain level. Right, thank you. Do we have another example? Okay, for example, about the, yeah, right, somebody talking? Do we have another one? Okay. For example, about the inertness of an excipient, magnesium stearate, which is the most widely used lubricant in tablets and capsules, it mainly may be used for 90% of the solid dosage forms. It have an incompatibility with acetyl salicylic acid, as magnesium stearate is alkaline, and over time it interacts with the acetyl salicylic acid, leading to its hydrolysis. So magnesium stearate, which is almost found in every preparation, is not absolutely inert. So understanding about the interactions that may be present between API and excipient must be understood, and make sure that even if this excipient is very widely used, it may be not suitable for these certainties. In this case, we may be using stearic acid, for example, that will be performing better along with the acetyl salicylic acid. For safety example, like the example we talked about of the excipient introducing nitrosamine, another example about safety is propylene glycol. Propylene glycol is a well-known excipient used in many formulations, but it is not safe for certain populations like pediatric. Their liver is not capable of metabolizing the propylene glycol efficiently, so it may accumulate in the body, causing toxicity. So propylene glycol must not be used for any preparations for the pediatrics or infantiles, but it is widely used for preparations for the adults. So the ideal properties of excipients of stability, safety, and inertness must be well understood, and the interactions that may appear with other ingredients or the API must be considered in order to get a stable formula over time. Main rules of excipient in the dosage form. We can talk about three main rules, which are industrial rule, biological rule, and patient compliance rule. Industrial rule, here the excipient is added in the product only to help its manufacturing in the pharmaceutical factory. And for example, the most widely used example of industrial rule are the lubricants. The lubricant is added to the tablets in order to decrease the friction between the particles of the powder to be compressed and the parts of the machine, like for example the punches that are performing the compression process. So these particles of the powder are not sticking to the punches and ensure a smooth compression process of the tablets. So here magnesium stearate does not have anything to do in the human body. It does not have a role for the product in the body, but it has a pure industrial rule. Its role is to enhance or facilitate the compression process of the tablets, or also for capsules when defilled to enhance its flowability on the machines, so the flowing of powder to be compressed or to be filled in capsule is smooth, so there is no sticking while compressing the tablets, for example. Another example is a biological rule, and here the excipient is placed in the dosage form in order to perform its action or effect in the body. And an example for the biological rule is a disintegrant. Here, a disintegrant is placed in the tablet in order to perform its function in the body, where it causes the tablet to rupture into small parts. These small parts will be easy to dissolve and release the API, so it can be absorbed to the bloodstream. So here the disintegrant is added in the tablet only to get to do its action inside the human body. That's a biological rule. Third is the patient compliance rule, for example, like coloring agents and flavoring agents for ODT, orally disintegrating tablets, or chewable tablets. So these are added to the tablet in order to enhance its acceptance by the patient and to have a better appeal while swallowing the tablet or while administering the tablet, so the patient will accept it without any problems. Another function may be mentioned also. It's a force function, which are stability rule. There are some excipients added to the product in order to keep it stable over time. Like, for example, the antioxidants and the preservatives. They don't have role in manufacturing. They don't have role in the body. They don't cause the patient to accept the dosage form, but the role is to keep the dosage form stable over time. So a fourth classification may be also included for a stability rule. Moving forward to the main examples of excipients used in solid dosage form. First, a filler or a diluent. Second, the disintegrant. Third, a binder. And fourth, a lubricant. We mentioned in brief the disintegrant and lubricant. So can someone discuss with us the effect or the function of a filler or diluent in the manufacturing of solid dosage forms? What does a filler or a diluent do in a tablet? Do we have an insight here? It's an easy one.
Please, can you repeat the question?
What is the function of a filler or diluent in the manufacturing of tablets?
To make up the bulk mass of the tablet.
Yes, right. Great. Exactly. Their main function is to create a bulk for the tablet or the capsule while filling or compression in order to be easily handled and to minimize any errors in the dosage or the amount of API reaching to the patient. If I have a tablet that has a concentration of 1 milligram or 2 milligram, we have to create a bulk for this very low dose in order to be easily compressed, easily handled by patient. And here comes the role of the filler or diluent. It is inert, it is stable, it is cheap. It is placed in the formula in order to create a bulk that is easily handled during manufacturing and during administration. Fillers or diluents may have other functions like enhancing solubility of the tablet or enhancing compressibility and flowability of the powder, but the main function of a diluent or a filler is to create a bulk. That is the main function of them. Second is integrant, and it has been discussed before that it helps the tablet to rupture into smaller parts, and these parts will easily dissolve and release their content of the API. And the third is a binder. Again, we are asking if someone can describe what is the function or importance of a binder in the tablet. That is related to its name, easy to expect. What is the function of a binder? Any volunteers?
It will bind particles together.
Yes, to help the particles of the powder to be compressed to adhere to each other, forming strong bonds between them, and a tablet is created. So binder is the contrary of a disintegrant. Disintegrant is added so that the tablet can disintegrate or rupture to small particles in the body, while the binder is added in order to make an intact tablet during compression process so it can withstand any further treatment or further processes like coating, like packaging, like transportation from a place to another. And then when administered, the disintegrant does its function and ruptures the tablet into small parts. Finally, the lubricant, like we talked about the magnesium stearate before, it is present to prevent the friction between the powder to be compressed and the machine parts, so it prevents any sticking that may occur during compression process. Main examples of fillers like microcrystalline cellulose, avicel, also mannitol and lactose, disintegrant like rospovidone, like cross-carbamylose sodium, binder like povidone or hydroxypropyl methyl cellulose, HPMC, and finally lubricant like magnesium stearate, stearic acid, sodium stearyl fumarate. Those are main examples of ingredients that you will be facing during a formulation process of a tablet or a solid dosage form. After talking about the main excipients used, we will have a glance about the main techniques used in solid dosage form preparation. And here we have two main techniques to have a glance about or to cover briefly, which are direct mixing and granulation. Direct mixing is a technique that will be always a first choice during preparation due to its simplicity. It's very simple. It does not have a high cost. It does not have the risk of failures or problems to occur during preparation. It does not require complex machines. It is the best preparation method to be applied if the characters of the powder will accept this, and we will demonstrate this later. While granulation is the second technique, which has further two types of dry granulation and wet granulation. And if direct mixing is not applicable or another aspect is required, then we will go for granulation process. Direct mixing here as simple as that all the ingredients are mixed together in a certain sequence. May be required geometric mixing if the quantity of the API is very small compared to the other excipients. And after this mixing, the compression process directly starts. So it is very fast, it is very economic, and very simple, does not require complex machines, does not require effort, but it has two main characters that must be present in the powder in order to achieve a proper direct mixing process, which are good flowability and excellent compressibility. Flowability is the ability of my powder to flow freely on the machine in order to fill the dies of the compression machine with the required weight and start compression of tablets. If the powder has a bad flow characters, then direct compression will not be efficient process to achieve a proper compression process. As if powder has bad flow, then the dies will not be filled evenly. There will be weight variation, and weight variation will lead to tablets that have the right amount of API. Other tablets will not be having the required amount of the API. So bad flowability will cause further problems during compression. And same for compressibility. If the powder has bad compressibility properties, then it will not be easily compressed on the tablet. We require very high compression force on the machines that may not be applicable, or may be applicable but will affect the machine. And after sometimes the machine will be failing to achieve this high process as the machine is affected by time and starts to wear out. So if the required powder to be directly mixed does not have the required flowability or compressibility characters, then we will be forced to go for another technique, which is the granulation technique. Powder flow must be assessed using different techniques like measuring angle of repose, measuring the flow rate, compressibility index, or Hausner ratio. All these are techniques used to assess the flowability of the powder. And if these techniques show a bad flowing powder, then direct mixing will not be applicable. Also for compressibility, if a powder requires very high force in order to be well compressed, then direct mixing will not be applicable for this powder. So moving forward to the granulation process, when to go for granulation? We may ask this question before answering it. Does anyone can suggest when we go for granulation instead of direct mixing? And here we know that we are going for a process that has complex machines and much more steps than just mixing and compressing the powder. So does anyone have an idea why we may go to granulation process?
First, if we have flow issue,
Sorry, I can't hear.
then first, if we have flow issue,
Yes.
and second, if API is inert to liquid, if we are using some liquid during for granulation, if it
Yes.
has not any effect on API, so we can go for granulation. And
Great.
third point is, if API concentration is very less, so we may face uniformity issue, dose uniformity issue.
Perfect. Perfect. Great.
Yeah.
Yes. There is reason to go for granulation, like the doctor mentioned, is a very low amount of API and also a very high amount of API. Extreme amounts of API always requires granulation because the API always have a bad compression and flow properties, especially if I am intentionally using an API of low particle size to enhance its dissolution rate and its dissolving. Then this API of very small particle size will always have, or in most cases, will have a bad flowability and compressibility. So for a tablet, if its weight is 1000 milligrams and it contains 800 milligrams of API, then this powder will not be able to be compressed directly, and granulation process is required to enhance compression and flowability. On the other hand, a very low amount of API will require granulation, as using direct mixing may not efficiently mix the very small amount of the API, and non-uniformity of mixing may occur, but granulation makes sure that the API of low concentration is very well distributed and evenly distributed in the powder to be compressed. Second is the modification of release profile, as on granulation, either dry or wet, created are granules of higher mechanical strength, and these granules will have a different release profile. So if I need to slow down the release of an API, which is directly compressed, we may try granulation, for example, dry granulation. So we achieve granules of higher particle size, of higher mechanical strength, and these granules may have delayed onset of release of the API, so we can match the release profile of the innovator product. So granulation, either wet or dry, will help me control the release profile of the API. And finally, to enhance compression and flowability, as for particle size of the granules, when the particle size is increased, this will have enhanced flowability and compressibility. But for a small particle size of granules, we will have less flowability and less compressibility. So when I start to granulate a powder, I can control the particle size of the resulting granules during the sieving process, and here I can control the flowability and compressibility of the powder that will be moving to the next step. We have two different types of granulation, dry granulation and wet granulation. Dry granulation is used for the powder that is sensitive to moisture or sensitive to heat. As always, after wet granulation, a drying process is required at high temperature to achieve granules of low moisture content for the product to be further stable. So if a powder or API is sensitive to heating or sensitive to moisture, then I must go to dry granulation. But for products that are not sensitive to moisture or heat, wet granulation will be applicable, and wet granulation creates granules of higher mechanical strength, so better flowability and better compressibility than the dry granulation. That's why we may go for some products to wet granulation only if it is acceptable for the API to withstand temperature and moisture. In some cases, if a product is sensitive to water, we may go for organic solvent to be used in granulation like isopropyl alcohol or ethanol. And here we can start drying at lower temperature than the temperature required to dry water. For dry granulation, we have two main mechanisms, which are slugging and roller compaction. And here, slugging is similar to a compression process. We use a high force press machine, which compresses the powder into large pills or large tablets that will be further sieved according to my requirements to produce granules of controllable particle size, and these granules will be recompressed again. But for a powder to undergo slugging, there must be certain level of flowability and compressibility in order to go a slugging process. If a powder has no compression properties at all, I cannot perform slugging, as the powder will not yield acceptable slugs of required force, and the process will be, we can say, in vain. So if a powder has certain level of flowability and compressibility, it can be dry granulated by using a slugging technique. But if not, we may go to roller compaction technique. Roller compaction is performed by using a device called roller compactor, where it has two rolls facing each other and rotating against each other. And while rotating against each other, powder is flowing in between them. According to the speed of these rollers and their distance from each other, this powder flowing between them will be compressed into a sheet. The properties of this sheet will be controlled by the speed of rollers, will be controlled by the clearance between the rollers, so we can get a strong sheet of high mechanical strength or a weaker sheet with lower strength granules. And then this sheet can be further sieved in a controllable manner to get granules of the required particle size and required properties. While for wet granulation, as we mentioned before, we'll be having a granulating solution, maybe water, maybe organic solvent, and this granulating solution is added to the powder while applying force using an impeller and a chopper, which are the components of the granulating machine. And then these granules are dried to required moisture content and then sieved to the required particle size that will achieve the required properties by the formula. Sieving process is milling the granules to the required particle size distribution, and as a general rule, as particle size distribution increases, better flow and compression appears, but slower release of the API. But as particle size distribution decreases, worse flow and compression, but faster release of the API. So a certain balance is required to be created in order to optimize the performance of my granules. I have to optimize the flowability and compressibility by increasing the particle size distribution, but at the same time, I have to optimize the dissolving rate or the release rate of the API by decreasing the granules particle size. So optimizing this process is essential in order to get a powder that performs acceptably on the machine, and also the tablet that performs in an acceptable manner when it is administered to the patient. Final process to check today is the process of compression. And here we talk in a simple way about the compression process. There are different types of compression machines, single press compression machines, and also rotary compression machines, but all of them will be having the same idea of compression and the same set of upper punch, lower punch, and die. And our final question for sharing today is, can someone briefly describe the mechanism of compression by using upper punch, lower punch, and die? What is the function of each one of these components in the compression process? That's our final question before the case study. So let's have an insight here. What is the role of the upper punch, role of the lower punch, and role of the die? in a compression process. Do we have an answer? Okay, simply, the die is responsible for the tablet shape, and it is the cavity through which the lower punch and upper punch will move up and down. According to the level of the lower punch inside the die, the weight of the powder to be compressed, which means the tablet weight, is controlled. If the lower punch is placed at low level in the die, then high space will be present that will be filled with high weight of powder, so higher tablet weight. If the lower punch is suited in a high level in the die, then it will have a low free space above it, so low tablet weight. And for the upper punch, according to its level from the die, we will control the applied force to the powder and hence the tablet hardness. If the upper punch is placed near the powder, it will exert high force, so tablet gets high hardness. If the upper punch is placed at higher level from the powder, it will exert lower force, so we get a tablet of lower hardness. After performing this compression process, in-process controls are performed, including hardness, friability, and disintegration. Hardness is the force required to be applied to the tablet in order to break. And here we are checking if the tablet can withstand further steps of coating and packaging and transportation from a place to another. It must have an acceptable hardness value. And this will affect the friability, which is the percent weight loss to number of tablets after certain number of rotations falling from a specified distance. Group of tablets are added to the friability machine, and it rotates 400 rotations for uncoated tablets, 300 rotations for coated tablets, and the weight loss of the tablets is checked after these rotations. If tablets are broken, if tablets are trimmed, if tablets lose more than 1% of their weight during this process, then tablets are considered friable, and we can conclude that they will not withstand the next steps of coating and packaging and transportation. So we must increase hardness value in order to get non-friable tablets. Disintegration is dependent on the hardness. As the hardness increase, the disintegration time will increase. If hardness decrease, disintegration time will decrease. Disintegration is the time required for the tablet to disintegrate or divide into small particles or fine particles at a certain temperature, 37.5 degrees, similar to the human body, at constant motion rate up and down. Monitoring this disintegration time will give us a clue about the rate of release of the API afterwards. So if we have a very high disintegration time, we must control the hardness value to get a lower hardness, so a faster disintegration time, or maybe we require a more major interpretation in the formula by increasing a disintegrant or decreasing a binder, for example, to get the proper disintegration time, and in the next steps, the proper dissolution profile. Also, it is important to understand the difference between disintegration and dissolution. Disintegration is the tablet to be ruptured into small or fine particles, but dissolution profile is the percent of API that has dissolved at time intervals like we discussed before. So disintegration does not include dissolving of the API, while dissolution is API dissolved at a certain time intervals. So that was a brief discussion about the formulation or an introduction to formulation of solid dosage forms, including a glance about the different types of tablets, including the formulation rule of selecting the innovator, preformulation studies, and formulation cycle, then the main excipients used in the manufacturing of solid dosage forms, main techniques of preparation, and finally a glance through the compression process and its in-process controls. And now we get to a case study where the release profile of a product is compared to an innovator product. As we can see that after 10 minutes, the reference product has released 78% of its content, while the generic has released 56%. After 15 minutes, 89% for the brand and only 69% for the generic. After 20 minutes, 95% for the brand one, while 81% for the generic. Then finally, at 30 and 45 minutes, the brand product has released all its content of the API, while the generic products has approached the end by releasing 91 and 97% respectively. So after the introduction we talked about before, I am expecting that someone may explain this case study and tell us what we can do in order to enhance the performance of our generic product. First, is this acceptable when compared to the reference product or not? And if not, what are the measures to be taken in order to enhance the situation? So I need a final volunteer to discuss these case studies. Do we have someone?
Okay, Dr. Abdurrahman.
Hello, doctor.
Hello.
I have just several questions first in mind.
Sure.
The generic product was made by direct compression or granulation?
The generic product for this case, let's say that it is performed by granulation.
Okay. Why I'm asking this is because I'm trying to exclude the content or the uniformity of the tablets, since it finished at 97, not 100%. That's why I'm asking, was it a direct compression one or a granulation? Being since it is a granulation one, I can see that it started quickly, not as quick as the innovator. But later on it slowed down and more. I'm expecting that it would need to add a disintegrant intra one for the granules to to explode or to disintegrate. So mainly I believe disintegrant should be added intragranular. That's the one I have in mind. I don't know if it's right or not.
It's perfect.
But the
Yes,
one
yes. Particles, granular particle size will play a major role here.
Sorry, I
Granular
can't hear
particle,
you.
granular particle size will play a major role here.
Yes, perfect, right. We can control the particle size of the granules to achieve a better release. That's a great insight. Yes, right. So do we have another suggestion? Two great suggestions seen now, adding a disintegrant, specifically intragranular, to help the granules rupture in a faster pattern. And another one is decreasing the particle size of the granules to achieve a more rapid release. Two great insights. Do we have another thinking?
Hardness will also hardness. We have to
Perfect.
decrease hardness also.
Perfect, right. This is the simplest way. Instead of changing the formula or changing the preparation technique, we may just require to decrease the hardness of the tablets, but provided that decreasing hardness will keep the tablets intact and away from being friable. In some cases, decreasing hardness won't be available as the tablets will become friable. So we must make sure that tablets are not friable after decreasing the hardness value. Great, thank you. Another suggestions? Do we have other insights? Okay, great. Like we said, possible solutions, you will find that you already mentioned some of them. Compress tablets at lower hardness, but make sure that tablets are not friable. Increase disintegration or disintegrant quantity. And if product is granulated, modify the sieving of granules to smaller particle size, but also put in consideration that flow and compressibility are not affected. And the fourth insight is we can decrease the quantity of binder. If the quantity of binder added in the tablet is very high, it may cause the product to be resistant to disintegration, even if a proper amount of disintegrant is added, as there is a limit for the addition of disintegrant. After a certain limit, it cannot be added. So we may require to decrease the quantity of binder in order to get a more soft tablet that is easy to disintegrate and release its content of the API. So that was all for today's webinar. And I'm thankful for you for attending this webinar. I hope it was informative. I hope you gained some knowledge about the formulation process through this webinar. And I'm extremely happy to have met each of you today. And this is a learning journey that we are starting together. I hope our learning journey does not end by just today's webinar. It's continuous together. If you access our websites, you will find another webinars that are released before and will be releasing again soon. Also, our courses available on the website regarding formulation and other subjects. So I am wishing to meet you again, and I am thankful to you for today. I hope you enjoyed it. I hope you gained some information, and I will be very happy to hear from your insights and your opinion about today's webinar in the future. Thank you very much.
Thank you, Dr. Min, for today's wonderful session. We appreciate the knowledge you shared. Thank you, Dr. Min. For everyone who enjoyed the session and wants to continue learning, the full foundation course is now available on our Pharmacology platform. The course includes 12 detailed modules covering everything you need to know about formulation from A to Z, with high quality content and real industry examples to deepen your knowledge. You can enroll using the promo code PharmaCourse10 to get 10% off, valid for the next 2 weeks. Don't miss the chance. And to download your webinar certificate, please visit our website after the session. And don't forget to share your certificate on LinkedIn and tag us. We love to see your achievement. Thank you all for joining. I am Tasneem Abdelghani, and it was a pleasure hosting you. See you next time. Thank you.
Thank you.